Chapter 6. The Sleeping Sickness Spectre, Trypanosoma's Stealth. Our exploration now leads us to Trypanosoma, a genus of kinetoplastid protozoa, several species of which are responsible for severe diseases in humans and animals. Perhaps the most notorious of these is Trypanosoma brucei, the causative agent of African trypanosomiasis, more commonly known as sleeping sickness, a devastating neurological disease transmitted by the bite of the tsetse fly. Another significant species, Trypanosoma cruzi, causes Chagas disease, prevalent in the Americas and transmitted by triatomine bugs, often called kissing bugs. These parasites are masters of evasion, capable of surviving and thriving in the bloodstream and, in later stages, the central nervous system of their hosts. Under the microscope, trypanosomes are typically elongated, spindle-shaped organisms, characterized by a single flagellum that runs along the length of their body, forming an undulating membrane. This flagellum, originating near the kinetoplast, a dense granule of DNA within the mitochondrion, whips back and forth, propelling the parasite through the viscous medium of blood and other bodily fluids with surprising speed and agility. Their constant motion and serpentine form can appear quite eerie, like tiny, restless eels navigating an internal sea. The life cycle of Trypanosoma brucei begins when an infected tsetse fly bites a human or animal, injecting metacyclic trypomastigotes into the bloodstream. These rapidly multiply by binary fission in the blood, lymph, and spinal fluid. In the early or hemolymphatic phase of sleeping sickness, symptoms include fever, headaches, joint pain, and swollen lymph nodes. If untreated, the parasites eventually cross the blood-brain barrier, invading the central nervous system. This marks the onset of the neurological phase, characterized by confusion, personality changes, slurred speech, seizures, difficulty walking and talking, and disruptions to the sleep cycle, leading to the characteristic daytime slumber and nighttime insomnia that give the disease its name. The creepy nature of trypanosomes, particularly Trypanosoma brucei, lies in their insidious invasion of the most protected sanctum of the body, the brain, and their ability to systematically dismantle neurological function. The progression from a seemingly manageable illness to a profound neurological disorder is terrifying. Furthermore, trypanosomes are notorious for their antigenic variation, a clever mechanism where they periodically change the proteins on their outer surface. This allows them to evade the host's immune system, as antibodies produced against one surface coat become ineffective when a new one appears, leading to waves of parasitemia and prolonging the infection. Chagas disease, caused by Trypanosoma cruzi, has a different but equally insidious progression. After an acute phase, which may be mild or asymptomatic, the infection can enter a chronic phase where parasites primarily affect the heart and digestive system, leading to potentially fatal cardiac complications or severe gastrointestinal problems years or even decades later. The silent, long-term damage inflicted by these parasites is a chilling prospect. Trypanosomes, therefore, represent a significant threat, microscopic invaders capable of inducing profound and often irreversible changes in their hosts. Their writhing forms a visual metaphor for the turmoil they create within. Chapter 7. The Flesh-Eating Fiend Leishmania's Mark. We now turn our attention to Leishmania, a genus of trypanosomatid protozoa that are the causative agents of leishmaniasis, a complex group of diseases with a wide spectrum of clinical manifestations. Transmitted by the bite of infected female phlebotomine sandflies, Leishmania parasites are intracellular, meaning they invade and multiply within host cells, primarily macrophages, the very immune cells that are supposed to protect the body from pathogens. This insidious strategy allows them to evade the host's defenses and establish chronic infections. The life cycle of Leishmania involves two main morphological forms. In the sandfly vector, they exist as flagellated promastigotes, which are injected into the skin of a mammalian host during a blood meal. Once inside the host, these promastigotes are phagocytosed by macrophages. Instead of being destroyed, they transform into non-flagellated amastigotes, a round or oval form, and begin to multiply within the macrophage, eventually causing the host cell to rupture and release more amastigotes to infect new macrophages. This cycle of invasion, replication, and cell lysis perpetuates the infection. Leishmaniasis manifests in several principal forms, the most common being cutaneous leishmaniasis, characterized by the development of skin lesions or ulcers, often at the site of the sandfly bite. These sores can be disfiguring and slow to heal, leaving permanent scars. Mucocutaneous leishmaniasis is a more severe form where the parasites spread to the mucous membranes of the nose, mouth and throat, causing extensive and destructive lesions. The most severe form is visceral leishmaniasis, also known as Kala-Azar, where the parasites invade internal organs such as the spleen, liver and bone marrow. If left untreated, visceral leishmaniasis is almost always fatal. The creepy attribute of leishmania is starkly evident in the physical disfigurement caused by the cutaneous and mucocutaneous forms. The idea of a microscopic parasite literally eating away at one's flesh is profoundly disturbing. The silent internal invasion of vital organs in visceral leishmaniasis, leading to systemic illness and eventual death, is equally terrifying. The parasite's ability to hijack the host's own immune cells, turning defenders into breeding grounds, highlights its evolutionary cunning and contributes to the difficulty in treating these infections. Found predominantly in tropical and subtropical regions, as well as parts of southern Europe, leishmaniasis affects millions of people worldwide, with impoverished communities often bearing the heaviest burden due to factors like poor housing, malnutrition, and lack of access to healthcare. The tiny, almost invisible sandfly, no larger than a grain of sand, becomes a formidable vector for this debilitating parasite. Leishmania, in its various guises, serves as a grim reminder of how microscopic organisms can inflict profound and lasting damage, leaving an indelible mark on both individuals and entire communities. Prepare yourselves, for we are about to encounter one of the most feared microorganisms on our list, often chillingly referred to as the brain-eating amoeba. This free-living amoeba is typically found in warm, freshwater environments such as lakes, rivers, hot springs, and even poorly maintained swimming pools or contaminated tap water. While infection is exceedingly rare, its consequences are almost invariably catastrophic, leading to a rapidly progressing and usually fatal brain infection known as primary amoebic meningoencephalitis, or PAM. Exists in three forms: a cyst, a trophozoite, the feeding infective stage, and a flagellate. The trophozoite is the form that causes infection. It thrives in water temperatures above 25 degrees Celsius. And infections most commonly occur when water containing the amoeba forcefully enters the nose, often during activities like swimming, diving, or water sports in contaminated warm fresh water. From the nasal passages, the amoeba migrates along the olfactory nerves, through the cribriform plate, and directly into the brain. Once in the brain, trophozoites begin to feed on brain tissue, causing inflammation, hemorrhage, and necrosis. The onset of symptoms is rapid, typically within 1 to 9 days after exposure. Initial symptoms may resemble bacterial meningitis and include severe frontal headache, fever, nausea, and vomiting. As the infection progresses, symptoms can escalate to stiff neck, seizures, altered mental status, hallucinations, and coma. Death usually occurs within 1 to 18 days after symptoms begin, making PAM one of the most aggressive and deadly infections known. The sheer horror associated with is undeniable. The concept of a microscopic organism actively consuming brain tissue is the stuff of nightmares. Its mode of entry, through a common recreational activity like swimming, adds a layer of insidious dread, turning idyllic summer pastimes into potential, albeit extremely rare, sources of lethal danger. The speed and severity of the disease leave little time for effective intervention. And despite some experimental treatments, the survival rate for PAM is tragically low, less than 4%. It is crucial to reiterate that infections with are very rare. Millions of people swim in warm fresh waters each year without incident. However, the extreme virulence of this amoeba warrants caution. Preventative measures include avoiding submerging the head or stirring up sediment in warm, stagnant fresh waters, especially during periods of high water temperature, and using nose clips or holding the nose shut when engaging in water activities in such environments stands as a terrifying example of how a seemingly innocuous, free-living microorganism can, under specific circumstances, become an aggressive and devastating pathogen.